For inviting me here today to give this talk. I will talk about increased damage to proteins in aging, wear and tear potentially avoidable by extending preventative maintenance of life's essential machinery. So there is a continual structural and functional impairment of the proteome in physiological system due to protein damage in vivo. And there are many types of damage, as previous speakers have mentioned. Oxidation of tyrosine, methionine and cysteine. Nitration occurs on tyrosine. Early glycation normally happens on lysine residues, and advanced glycation and products are formed on arginine and lysine. This type of damage increased in aging and also in diabetes, renal failure, arthritis, and many other diseases, and may be related to condition progression and remedial intervention. So there is a family of oxidation addicts. Cysteine, methionine, tyrosine, and tryptophan can oxidize. These are the chemical structures, and there are family of glycation addicts include early glycation addicts, fructosyl lysine and fructosyl amino acids, and lysine-derived AGEs and arginine-derived AGEs, CML and CEL, probably all aware, are minor AGEs. The most quantitatively important advanced glycation is arginine-derived hydroimidazoles. There are cross-links, many mold and glucospan, and some are fluorophores. Pentosodine and arginine are well studied because of their characteristics of fluorescence characteristics. So there is a continuous process of damage to the protein in tissues. Protein gets damaged by these three processes and then leads to proteolysis. Proteolysis product is leaked out in the plasma, where it filters in glomerular filtration, glomerulus. Some of these damaged amino acids can be actively reabsorbed, and some can be excreted and eventually excreted in the urine. There is a controversial area of food. Processed food get the protein in that can be glycated or damaged to the higher extent, and most of that damaged protein is excreted, but some of the lower damaged protein can be digested, and the digested peptide are then through portal vein released into the circulation. So there is a nomenclature of describing all the process which I've shown. The protein damage, when we measure these protein damage addicts in the protein, these we refer to oxidation, nitration, and glycation addict residues of protein. Of peptide measurement is called addict residues of peptides, and the debris which we free of amino acid, damaged amino acid proteolysis product is normally referred to the free addicts of the damaged amino acids. So we have developed a MSIMS analysis assay to measure all these protein damage markers. Processing of the sample is done. Protein is delipidified by ether or methanol chloroform extraction, and protein is washed by ultrafiltration. Then series of enzymes are employed to digest the protein into component amino acids before analysis. This is now fully automated process. We use LC-MS tandem mass spectrometry, and the internal standardization and calibration is achieved by standards and stable isotopes, substituted standards, which we have synthesized in-house. Now there is a company in France can sell some of the these structures I have shown you previously. We use hypercarb graphitic column. This allows us to inject the digested protein directly and preventing any, which allow us not to derivatize the sample and prevent any interference. Analytical performance of this assay is the limit of detection for ages and amino acid is 5 to 80 femtomole. Recovery is greater than 80% for AGEs and 94 to 100% for amino acids, and inter-batch variation is less than 10%. So these are specimen chromatogram of the mass spec. These are the glycation, oxidation, and nitration marker. This is the internal standard, and this is the peak showing in the test sample. And this is tryptophan, oxidation marker, NFK, again internal standard, and the test sample. This work was done in collaboration with Professor Linda Partridge and David Jem's group, and still we have to publish this work. So developing this method, we are able to quantify about 25 chemically defined oxidized, nitrated, and glycated amino acids. So by measuring these free addicts in urine tells us the flux of damage of the physiological proteome, and damaged amino acid residues of protein are linked to functional impairment, susceptibility to damage, and repair and replacement. So we have, over 10 years, measured all of these markers in different diseases and aging, and we have a pretty good picture of which kind of damage is really, or really there are many these 4 types of damage are important in disease process, not just oxidative stress. So glycation by dicarbonyls are the most important damage occur in physiological system. Methylglyoxal is an extremely reactive compound. It reacts to, directed towards the arginine residues, reacts and form hydroimidazole, advanced glycation end product. So where this methylglyoxal come from? Methylglyoxal is produced from triose phosphate degradation through glycolytic pathway, and normal flux is 0.1% of the triose phosphate degraded to methylglyoxal. It is also formed from ketone body metabolism and threonine metabolism. In hyperglycemia, GAP3 pH level goes down, increasing triose phosphate and consequently increasing the flux, degrading to methylglyoxal to 0.2 to 1%. But there is a protection against the glycation, and that is the glyoxylase system. It protects against glycation by methylglyoxal in physiological system, protein damage, or DNA damage. So glyoxylase is, this slide shows the timeline of glyoxylase. It was discovered in 1913, but really for aging research, it came into, in 18th and 19th, it was discovered that the GLO1 and GLO2 activities decline in aging human length. And in 2000, there was shown that the MGH1 is a major type of protein damage in vivo, and Michael Morkos published that the GLO1 is a beta gene. And again, copy number variants of human GLO1 was discovered, and in mouse, and methylglyoxal-derived DNA damage paper was published in 2010. So is there evidence that glyoxylase 1 protects against glycation, aging, and influence lifespan? Again, a group in Heidelberg, they studied the GLO1 in C. elegans and found that it declines with age, so this is the mRNA of GLO1 in C. elegans in 12 days old, and found there was about 60% decrease in mRNA level. Same group then engineered overexpression of glyoxylase-1 and studied the effect on aging, and they found that there was overexpression of glyoxylase-1 in C. elegans increased median and maximum lifespan by about 30%. And silencing of glyoxylase-1 was a decreased lifespan by 40%. We measured the glycation markers in the study group and found that the fructose, lysine, CML, and 3-DGH, there was no change. So there was no effect on glucose-dependent glycation and related degradation product-derived AGEs. We also measured methylglyoxal-derived AGEs, CEL, MGH1, and GH1, and as you can see that these were decreased in older transgenic C. elegans compared to the wild type. We also found in these groups that methionine sulfoxide is quantitatively a major oxidation marker and nitration marker through nitrotyrosine were also decreased in GLO1 transgenic C. elegans compared to wild type. So overexpression of GLO1 suppressed oxidative and nitrosative damage to the proteome, but the question is why? To understand that, we looked at the mitochondrial protein for MG modification, and to quantify this slide, we found that the transgenic GLO1, transgenic 12-day-old C. elegans have lower MGH1 level in mitochondrial protein compared to wild type, and silencing of GLO1 increased it significantly, leading to increased superoxide formation and oxidative damage is possibly. Then we looked at the ROS formation in these study groups, and again quantifying that, we found that again transgenic C. elegans have lower ROS production similar to these wild type when added uncoupler protein agent, mitochondrial uncoupler protein agent, and on silencing, the ROS production was increased significantly. So what we know from this study that enhanced protection against dicarbonyl glycation increases the lifespan of C. elegans, and decreased protection against dicarbonyl glycation decreases the lifespan. Glycation by methylglyoxal is non-oxidative but can stimulate oxidative and nitrosative stress. So dicarbonyl damage to the proteome may be a further key component of the aging process. So we do need to study dicarbonyl stress. We have shown that the proteins susceptible to dicarbonyl glycation and functionally important sites normally are albumin, hemoglobin, crystallins, mitochondrial proteins, extracellular matrix protein, collagen, fibronectin, and laminin, SYN3A and integrins, and many, many more. There are sources, many sources of these glycating species. There are exogenous and endogenous sources. Exogenous sources are smoking, thermally processed food and beverages, ultraviolet light, and ionizing radiation. Endogenous sources, monosaccharides, trisphosphates, ketone bodies, and lipid peroxidation, but of course there is anti-glycation defense, glyoxylase-1, aldehydo-reductase, and others. So methylglyoxal is the most reactive, and glucose is the least reactive glycation agents. Methylglyoxal have the ability to impair physiological function of protein by modifying them at functionally important sites, and consequently causing aging, disease, and cell death. So can we intervene in aging process and possibly reverse it to rejuvenation? Obviously everyone is trying, but probably not. But what we can achieve is healthy aging. This is Professor Tonelli in 1992, and this is 2008. I can't see much difference. Maybe he knows something what we don't know. So healthy aging can be achieved by activating NRF2 system. It is a little bit scary slide, but I would like to just try to simply explain. NRF2 normally complexed with KEAP1 and other accessory protein, and is always led to proteasome for proteolysis. By endogenous activator or exogenous activators, dietary bioactive compounds can disrupt this complex, and NRF2 is then phosphorylated and translocated into the nucleus, where it combined with other MAC protein and accessory protein binds to ARE promoter region and increase the expression of protective protein and decrease expression of lipogenic proteins. It is a complex process, and we have recently discovered that GSK3 beta and fin kinase are important. These fin kinase phosphorylate NRF2 in the nucleus and expel that from nucleus into the cytoplasm. So it's kind of turning on by these activator and switching off system exists, but there is still more to learn about it. So by activating NRF2, we can have protection against an oxidative damage by increasing the expression of all of these antioxidant enzymes, protection against metabolic stress, protection against lipogenic stress. It downregulates steroid response, alanine binding protein 1 and fatty acid synthase. And if it all fails, it also increases proteasome, in 20S proteasome and sequestosome for removal of the damaged proteins. Last but not least, there is by activating NRF2, we can also protect against glycation. Again, we found last year we published that the glyoxalase 1 also is RE-linked gene, so it is also activated by NRF2. This slide shows the activation of these time course of mRNA levels in HMAC1 endothelial cell stimulated by sulforaphane. Sulforaphane is a bioactive, dietary bioactive found in brassica vegetable. So as you can see that the antioxidant enzyme were induced, anti-glycation enzyme, anti-metabolic enzymes, and it also regulates NRF2 and KEAP1, and inflammatory related genes were downregulated, NF-κB and VCAP1. We found that GLO1 is induced by sulforaphane, but it is a complex system. We found not every bioactive can induce every gene, so different bioactive work differently. So glyoxalase 1, if quinone reductase or antioxidant enzyme is increased by one dietary activator, it doesn't mean you will get glycation and anti-glycation enzyme will induce with the same. So it's still a lot to understand. So this heat map shows that about 50 different genes upregulated and downregulated in HAPG2 cells. Lipogenic genes expression was declined while anti-glycation, oxidation defense genes were increased. We measured the protein damage marker in these cells, stimulated and unstimulated, and found that methionine sulfoxide and MGH1 was decreased in sulforaphane-treated cells compared to the control group. Again, we found that in human fibroblast senescence studies that NRF2 and transketolase decline as the doubling of the cells. So for healthy aging, we have to correct the decline of the NRF2 and the expression of AR-linked genes. And this can be achieved by dietary, by these vegetables, by eating fruit and vegetable. These are rich in NRF2 activators, and that is what we are working on. Actually, we have found a partner with we form a partnership with Unilever to prepare a functional food for vascular health. To summarize, protein undergo continual spontaneous modifications in physiological system, increasing with age. Damaged protein undergo proteolysis with excretion of damaged amino acids. Accumulation of protein damage in aging occurs as a consequence of increased rate of protein damage, probably due to decline of anti-defense of all the enzymes, decreased rate of repair of damaged proteins, and decreased rate of proteolysis of damaged proteins. Oxidative and non-oxidative mechanisms are involved. Healthy aging may be available for people through dietary supplements which prevents decline in battery of gene, protected against multimodal stresses by activation of the anti-stress gene response. With that, I would like to acknowledge this is Paul Tonelli, co-director of Protein Damage and Systems Biology Research Group with me, and our team. I would like to thank BBS ASSAY and Wellcome Trust and FP7 for their support for our research. Thank you very much.